All right, so chapter 6 begins our talk about the cell. And in 1657, when Van Leeuwenhoek was kind of writing about what he had discovered with his microscope, he wrote, "No more pleasant sight has met my eye than this of so many thousands of living creatures in one small drop of water." So, what looked to be pure water, when looked at under a microscope or magnified greatly, revealed that there was lots of living organisms in there. And that was kind of what got the whole idea of microscopy and microbiology and that type of thing going, was when Van Leeuwenhoek, who was largely considered to be the father of the microscope, really invented a way to look at things that aren't visible with the unaided eye. Okay, so some of the essential ideas in this topic here are the evolution of multicellular organisms allowed cell specialization and cell replacement, and eukaryotes have much more complex cell structure than prokaryotes. And from the TOK perspective, what I need you guys to do is to take and write a small paragraph on this. There is a difference between the living and the non-living environment. How are we able to know the difference? So take a few minutes, think about that, write something down, and then we'll make sure that we talk about it in class. And then go ahead and do the same thing for this. The world that we inhabit is limited by the world that we see. Is there any distinction to be drawn between knowledge claims dependent upon observations made by sense perception and knowledge claims dependent upon observations assisted by technology? Okay, so again, write a paragraph for each of those and then be ready to discuss this in a either a Socratic type of a format or just kind of in general where we share out. Okay. All right, so cell size. The cells, of course, we said in class, are the smallest living entity, and there's lots of different types. You have simple bacteria, you have single-celled organisms, you have complex multicellular organisms, etc. So there's all different types of cells that contribute to living types of organisms. And we said also that if you kind of look at things a certain way, certain properties begin to emerge just depending on how you look at them. And we said to emerge means to like come out of, okay? So it's important that you keep in mind that multicellular organisms definitely have the idea of emergent properties in them. Because if you look at it, the whole thing is much greater than the sum of the parts. So the individual pieces that make up the large multicellular organisms largely can't exist on their own. Things that make up the cells largely can't exist on their own, but that entire organism can't exist without those small parts either. Okay, so when you start looking at certain things, for instance, using a microscope, you can see with the unaided eye, you've got a leaf. And you might be able to even look at that leaf and see certain structures like the veins and perhaps some of these small branching types of things that kind of emerge out of it. But as soon as you throw that under a microscope, what you start to notice is that the leaf has a lot of types of emergent properties. In other words, okay, the green organelle is called chloroplast. There's certain cells, certain structures within those cells, cell walls, etc., the nucleus perhaps, and so forth. And if you zoom in further on these chloroplasts using a different type of a microscope, an electron microscope, you can see that the structures that make up the organelle really start to reveal that that substructure within the cell itself consists of small components that the organelle, the chloroplast, could not work without. Okay, and then when we start to get into the cells, plants, photosynthesis, we'll talk about some of these structures within the chloroplast, for instance, like the thylakoid membrane, the stroma, and all of the different types of things that are making up the organelle that we're talking about here. And if you could zoom in even further, you would find out that there's molecules and, of course, atoms that are making up those particular structures and so forth. Okay. If you zoom out a little bit and you start looking at the leaf, you might notice that it's green, okay, that it was attached to a branch and that branch was attached to a tree, etc. So just kind of your perspective for looking at these things often reveals different things about them that you might not otherwise notice if you weren't kind of looking at it a particular way, okay, and that's really what the idea of these emergent properties is. All right, so now, as far as living matter goes, living matter is made up of carbon, hydrogen, oxygen, and nitrogen. That's the bulk of the living matter. That's about 96% of it. And then we said in class the remaining 4%, generally speaking, is phosphorus, sulfur, calcium, and potassium. All right, and then there's a few trace elements, and these elements are required in extremely small quantities, and this is what helps your body really function in a normal way. A lot of these things are cofactors that exist in the functioning or exist to help the enzymes function, perform their jobs, help your body kind of maintain its health, healthy immune system, operate within that kind of narrow range of homeostasis and so forth, etc. Okay, and we went over the idea of some of the trace elements being like selenium or manganese, magnesium, iodine, etc. And those are some examples of them. And there's more in your book, and we'll talk about more in class certainly. Okay. So as far as microscopes are concerned, these were generally invented in the 1600s, and they're using light in some way, shape, or form to view a specimen. Now the ones that we use, of course, use a light bulb, but back then they were just using like reflected light with a mirror or like a light colored background and so forth. And in some of the videos that we look at in class, you'll see that these microscopes probably don't resemble anything that you're really visualizing in your head right now based on what your knowledge of what a microscope actually is and looks like, okay? Those light microscopes were used for a few hundred years, and then as technology advanced, as is often the case in science, when technology advances, science advances, and so forth. So the technology brought us to the idea of being able to use an electron beam to create an image of a structure that had been prepared a special way, and using what's called or what's known as an electron microscope. And these were invented in the 1930s to look at non-biological types of materials, for instance, like metals and small parts, electronic parts, etc. And then someone had the idea that if we take these living specimens and prepare them a certain way, which unfortunately kills them, but prepare them a certain way, we can visualize them with an electron microscope and reveal all sorts of crazy detail that we wouldn't otherwise be able to see. So some of the advantages of light microscopes is that we can easily view slide preps up to 1,000 times what their normal size is. We can view living organisms with light microscopes, and they have a wide variety of types of uses. And then some of the disadvantages is also that, for instance, we can only look at it up to 1,000 times, whereas with an electron microscope, we can look at it, many more thousands of times greater than that. The limitations in terms of the resolving power, and we'll get into that really more in just a minute. The limitations of the resolving power is also one of the disadvantages. With the electron microscope, some of the advantages are that they have a great resolving power. We can really see the great fine detail of these things. There's a wide variety of uses. Two of the very common ones are scanning electron micrograph or a microscope where you're looking at the surface of an object, or a transmission electron microscope where you can actually see through these things. And the other advantages is that they have tremendous resolving power, down to about 1 to 2/10 of a nanometer, or 1,000 times greater than that of a light microscope. The disadvantages are the microscopes themselves are very expensive. They're hard to maintain. Usually you only find these things at like a university or certain businesses, things like that. Slide preparation is very costly, very difficult, because you need to use electron dense types of elements like gold and osmium are two common ones that you would use to kind of coat the surface of an object to be able to reveal the structure that you're looking at and so forth. One of the other disadvantages is the process to prepare the specimen for study definitely kills it, and therefore you can only look at living or dead organisms, whereas with light microscopes you can look at living types of organisms. Okay. Now, as far as resolution is concerned, we talked about the resolution of a light microscope back here, about 2/10 of a micrometer, and with the electron microscope we said the resolution was about 1 to 2/10 of a nanometer, and that resolution is basically the distance between two objects, being able to see them as separate and distinct objects. So for instance, let me switch the color here. Okay, so if we look at this right here, okay, you can see that the two objects as separate and distinct objects. And the resolving power is that distance that you would see between these two objects. Okay, So obviously the distance here is much smaller with an electron microscope than it is with a light microscope. Okay, so what you can do with a high resolution is determine whether this object that we're looking at right here is actually kind of this weird shaped blob, or if it's actually two dots that are separate and distinct. So the higher the resolution, the more detail you can see. And really what you look at here, if you look at the figure on the right, if we think about the wavelength that we're looking at things with, okay, this wavelength here of the light would be, for instance, like a basketball. Okay, so we have a large wavelength here, say with red light, okay, and then hopefully you remember from like your physics class, as you get to like violet light, the wavelength becomes a lot shorter. So this could be blue or violet light, and those are the blue light is ideal for looking at it because that would be like using these jelly beans. So when you have this shorter wavelength, you have a higher resolution versus having a longer wavelength. Okay, and when we use electrons as the beam instead of visible light, so this over here, this would be visible light, and this would be like in a light microscope. And then if we used these beads, this would be like an electron microscope. And these electrons have a very, very short wavelength, which is why we get such a very, very high resolution right here. Okay, very high resolving power. Okay, so if we look at, for instance, this here's the head of a pin. You get poked with a pin, you know, you're supposed to clean it good, sterilize it, and that's because usually when you're poking yourself with something, it's not sterile. And so if we look at the head of this pin right here, this is just at 91x. Okay, this is like with a magnifying glass almost, you know, just a it's not, but you know, you can see the tip of it. But when you zoom in with an electron microscope, you're starting to see that it's crawling with these bacteria. So you can see now here, down here, it's you know, over 12,000 times blown up. You can see the shape of the bacterium, for instance. So if you stick yourself with a clean pin, if it's not sterile, you're probably poking yourself with some bacteria or something like that. That's what we're showing you here. Okay. So some of the size ranges of the different things that you can look at. For instance, if you look at this with a transmission electron microscope right here, you can see that you're getting right down to the molecular level, really on up to really the size of about an insect. And then with the scanning, it doesn't have quite the same resolving power as the transmission scope, but again, you can get down really to the molecular level when you're looking at certain types of viruses and organelles within the cell and proteins and things like that. With the light microscope, the thing that we're largely used to, this is where you can see things down to small bacteria up to, of course, with looking at insects and things like that. And then the unaided human eye, really you're able to see things that are very, very large cells, for instance, like the human egg that would be released during ovulation. That's about the size of the period at the end of a sentence in your textbook, for instance. So you can see that with the unaided eye, obviously all the way up into large objects. So that's kind of the size range of cells. Okay. So as far as the microscope is concerned, there's a lot of people that were working on microscopes at the time. And, you know, there's no real true father, although we do like to talk about Van Leeuwenhoek as being considered to be the father. But a lot of people really wanted to take credit for this. We have the Jansons here. They made the self-proclamation that they were the fathers of the microscope. And you know how that goes when people are usually braggadocious. There's usually a lot of you know, falsities associated with that sort of a thing. Galileo is another person who was working on lenses, and of course we know Galileo really working with telescopes and so forth, but he made a microscope that had two lenses as well as the telescopes that he did, but they had a lot of aberration, and really what aberration means is like distortion. And if you've ever looked like sideways through a piece of glass, you can see that it kind of gets bent and you get a rainbowing effect and so forth. And that would be considered to be an aberration. That rainbowing effect would be chromatic aberration. And then Van Leeuwenhoek, he's the guy that really did a lot with microscopes. He was very prolific in terms of the numbers that he made. You know, he made many hundreds of microscopes. And one of the problems with him, though, is that he was very selfish. He didn't share his knowledge or his ability to do these types of things, and therefore he kind of held back really the idea of microscopy and biology and that sort of a thing, because he didn't share really what he was able to do with this. And he was kind of a master craftsman in terms of making these lenses, because what you have to do is you have to kind of take the piece of glass and round it. In the more perfectly round it becomes, the higher the resolution you get and the less aberration that you get from these types of things. So many of his scopes were about 250, 260 times, or had the ability to blow things up about 250, 60 times. Other types of lenses could magnify things over 500 times, okay? And some of these things were just giant lenses. Other things were kind of hooked up to a little metal device where you could kind of look at and see different things. And in some of the videos that we'll watch in class, we'll be able to see these. Another man named Christian Huygens, what he had the ability to do was really correct these lenses for their chromatic aberration. So he had ability to make them really smooth and round and really get rid of a lot of the distortion that's caused by manufacturing these types of things. So the idea of the cell comes from a man named Robert Hooke, and what he was looking at in the 1600s was these slices of cork. And these slices of cork remind him of a monk's residence, where they live in these things that they were called cells. So he kind of gave the idea of these living kind of things, so to speak. He called them cells. And he wrote a book called Micrographia, and it contained a lot of detailed drawings, and we'll look at some of the drawings in a video that we'll watch. And all of these things that he was seeing under the microscope, he had the ability to really draw them, and these were things that people had never seen before. So there was a lot of interest in these types of things. And his book that he wrote, Micrographia, was considered the first book of microscopy, and this is what people were able to marvel over when he showed them to them. So the idea of the cell led to the notion of cell theory, and this is really kind of an evolving type of a theory. And Schwann and Schleiden were two people that were responsible for coming up with this. And basically, the 5 things that they talked about here are things that we should know. Okay, so what they said was that cells are the fundamental units of all living things. The cell is, whether it's one cell or many cells, if you're living, you are comprised of at least one cell. And all cells come from pre-existing cells. So when they grow and divide and give rise to new ones, that's where the idea of cells come from. And the genetic material gets passed from one cell to the next during the copying, and that's how these cells are able to live from one generation to the next. And more or less, any type of cell, no matter what you're talking about, is the same chemically. Whether it's a bacterial cell or a eukaryotic cell, they all have certain fundamental things that are the same throughout them. And then what they have that's different really makes them different. But all cells have like a cell membrane, they have cytoplasm, they have genetic material, they have ribosomes, and so forth. And so essentially, they're all the same, despite the many differences that we probably know about. So there were some other people besides Schwann and Schleiden that were working on this. Robert Remak and Rudolf Virchow were 2 people. They made significant contributions to the idea of cell theory, and they were initially friends, but they had a falling out because what Virchow wanted to do was really kind of take this work and make it his own, and really be the guy who came up with it, and not share the fact that Remak had a lot to do with it as well. And some of the kind of problems with this was that Virchow was a German person. He had a lot of friends in politics. He had a lot of influence over kind of the sphere that dealt with cells and so forth, as well as many other things. And Remak was a Polish Jew, and he basically was kind of pushed into obscurity because of Virchow and his kind of political standing, his societal standing, etc. And really until recently, most people had not heard of Remak and didn't know that he contributed a lot to the idea of cell theory. So in the video that we're going to take a look at, we'll be able to see how these two kind of interacted and what some of the ramifications for what Virchow did were. Okay? So as far as like really the general knowledge at the time, the idea of spontaneous generation, most people were highly uneducated at this time, and they just basically believed in things that they saw. So in other words, worms came from meat, okay, rotting meat, maggots in the meat. They just figured that the worms came from the meat because a lot of people weren't really doing a lot of observing at the time. They saw something and then they saw it again and there was a change, and they didn't really have the education or the understanding to know where this stuff came from. Beetles came from dirt. Rodents came from grain, etc. So there was a lot of experiments going on by scientific people at the time. That didn't support the idea of spontaneous generation. But the problem was is most people were uneducated and they still believed in this idea. Okay, so a couple of the experiments, you had Francisco Redi and Lorenzo Spallanzani, and they were doing some experiments to kind of disprove this notion of spontaneous generation. And so what Redi was doing was he took this jar and he put some meat in it, and then he made some observations, and he noticed the flies were going in and out and landing on the meat, etc. And then they left. And then eventually the maggots, the worms came from the meat, and his conclusion was that the flies must somehow be involved. So he prevented the flies from getting there by putting some cork over the piece of meat so the flies couldn't have access. But the thing was is the people didn't believe him. What they said was, well, there's no air, so that's why the worms can't exist. So he said, okay, yeah, fair enough. So what he did was he put some cheesecloth, which is, very porous, but doesn't allow the flies through, but allows the air in and out. And what he noticed is when he put that over the top of it, the flies didn't have access to the meat, but they did have access to the top of the cheesecloth, and you could still have the odor. So the flies landed on the cheesecloth, they laid their eggs, and you'd get the maggots at the top of the thing, but not in the meat. So he was able to show that these flies were giving rise to the maggots, which were giving rise to more flies, etc. Lorenzo Spallanzani was another guy. What he did at the time, when you wanted to kind of preserve food so that you could eat it in a few days later because they didn't have any refrigeration or anything like that. He showed that when you boiled broth and then you sealed it immediately, there would be no growth because things couldn't get in from the outside. And when you boiled the broth and left it open to the outside, it would turn cloudy. And so, that experiment that Spallanzani was doing was kind of furthered by Louis Pasteur, and the whole idea of what these people were doing lent a lot of support to the idea of cell theory, because what they're showing is that these cells were coming from pre-existing cells. So what Pasteur was able to do was use these swan-necked flasks, and I'll show you, they kind of look like this, okay? And so what he was able to do was he was able to boil off this fluid, and then obviously the gas could escape out through here, and the air could get back in, but anything that was in the air was kind of trapped right here. And so what he was able to show was that when you boil these things and you don't allow anything access to that, then it's not going to spoil. And then what he was able to do is he was able to take this device and kind of tip it and then wash some of the fluid down into this region, collect the dust and the sediment that had settled there, and then when he tipped it back up, some of that stuff that was trapped in there kind of made its way back into the solution, and then within a couple of days it started to turn cloudy. And so he was able to show that spontaneous generation really wasn't what was happening. What was happening is things in the air and the atmosphere, organisms, etc., were getting in, and that's what was causing the spoiling of the food and so forth. Okay, so once we got rid of the idea of spontaneous generation, what Robert Koch was able to do was kind of come up with this notion of germ theory of disease, establish the idea that, if you're sick, you have the ability to get other people sick because of the organism that is in you getting you sick gets transferred to another particular person or whatever, and then they become sick as well. And this gave rise to the notion of Koch's postulates. And what he was basically able to show is that the specific agent of the disease has to be present in the person or the organism with the disease. In other words, if you have strep throat, you need to have that streptococcus bacterium in you, or else you don't have strep throat, you've got something else. And so what you can do, for instance, if you think you have strep throat, you can do the throat culture where they stick that little swab down your throat that makes most people gag, and then you kind of plate it out onto a piece of, an agar plate that usually has like sheep's blood in it. And if the streptococcus bacterium is there that causes strep throat, it's going to lyse the blood cells that are in there, and you're going to get these little colonies that grow with the kind of ring around them where the sheep's blood has been destroyed. And then if you take, and this isn't something that you would do, but back then things were a little different. If you take the bacterium that causes strep throat in this person and you inoculate somebody else with that, they must also come down with strep throat, okay? And when they come down with strep throat and you swab their throat and you plate it out on an agar plate, you see that they have the same organism that was making the initial person sick is now making them sick. And that's what is referred to as Koch's postulate. So you can kind of see that cycle of what's going on. The cells, the bacterial cells in this particular example that are giving person A strep throat are also giving person B strep throat. And those things have to be isolated as well to kind of confirm that that's what you have. So that's kind of what Koch's postulates are, okay? So with light microscopy now, just kind of going back to that, you could look at these types of things that are making certain people sick, the bacteria and so forth. We can look at them with microscopes now. And when you're looking at things, we speak of magnification. And this is what we did kind of in our lab when you multiply the objective by the eyepiece or the ocular, and then that's showing you how much it's blown up compared to real time. We talked about the resolution or the clarity of the object, that minimum distance between the two. It's much greater in an electron scope than it is, say, in a light scope. So we talked about those. And the idea of resolving power, and this is a little bit out of order, I apologize, but you can go just kind of add it to that part. But your resolving power, you don't need to know necessarily the equation here, but what I want you to understand is that the wavelength of light has something to do with it. So the higher the wavelength that you have, really that's going to kind of reduce your resolving power. It's going to make, how do I want to say this? Like, I guess if you don't use the correct wavelength of light, then your resolution isn't going to be as good. Blue light is typically the best for light microscopes because it's got the smallest wavelength, and that's going to give you the highest resolving power. And generally, too, when you switch objective lenses, the numerical aperture is the amount of light that you can allow in. And we showed you in class that when you switch objectives, your light gathering ability is going down, and that's what's increasing your resolving power. In other words, you can see a finer detail with that smaller scope, with the smaller aperture, as well as that wavelength staying the same. Okay, so generally speaking, the blue light is the better light to use to look at things under the light microscope, because the blue light has a small wavelength, so that's going to increase your resolution, okay, because your aperture is going to change with those different objective lenses, okay. Electron microscopes have a very, very small wavelength, okay, and a very small aperture, and that's what's going to allow that high resolution that you wouldn't get from a light microscope that you do get from the electron scope, okay. So next time, I got one more thing here to go through just to show you real quick. With the scanning electron micrograph, you can see here there's the object on the left right here. That's an earthworm. Okay, so you can see the fine detail there when you blow it up. And then over here you can see, these are different kind of organelles right here. We've got smooth and rough ER, and you can see the actual ribosomes that are studying the membranes right here. Okay, and then over here, of course, they're not, but that's the smooth and rough ER. So this is the fine detail of the interior of that cell. Okay, all right. So next time, what we're going to go through are the two main types of cells, and we'll talk about some organelles and things like that. Okay, so if you have any questions, make sure you write them down, and we'll be sure to answer them in class. Okay, we'll talk to you soon.